of our olfactory bulb, but we are going to look at that slightly later on. So from our primary vesicles, we have secondary vesicles, and then we have our neuroepithelium. Within the mesencephalon, although it doesn't develop much, we now develop some swellings of the mesencephalon. These four swellings that are referred to as the corpora quadrigemina. Corpora quadrigemina, superior and inferior colliculus. When we look at the brain, we'll be seeing these hump-like colliculus that we refer to as superior and inferior colliculus. And we're going to see how this comes about. This is just the same structure, noting the flexures and also the development of the three germ vesicles. During this development of the telencephalon and the others, you will notice that the telencephalon grows very fast. As it grows very fast, we have this type of growth where the cells spread out, the most close cells here spread out. And here we have this where the cells grow like in an inverted C. This is what we are calling hemispheric rotation. Hemispheric rotation. So during this hemispheric rotation, this part here comes to be buried by the telencephalon. Similarly, when you look at this, our mesencephalon and diencephalon will be almost buried by the telencephalon here. So this creates this part here, which we refer to as the insula, which is an older part of the development of the brain. Again, there is that process of proliferation and migration of the cells. Proliferation and migration of the cells. I'd like us at this stage to note that the brain, cerebral cortex and cerebellum, what you see on the outside is white matter. Sorry, gray matter. And what you see at the interior is white matter. This is to increase the number of neurons without increasing the size of the brain by having gray matter on the outside and white matter on the inside. In the case of the spinal cord, the white matter is on the outside, the gray matter is on the inside. So how has this come about? That's just what we are going to look at now. This was just an illustration of what we are talking about in terms of this is the tip of the crest where you have the neurocrest, the crest of the trench, and this is now where we have the neurocrest cells. This is our trench, this is the fold, neural fold, and this is our notochord inducing this. I've said two things. Once this fuses, we have migration of the cells at the crest as neurocrest cells. Some of those cells do not move very far, remain very close to the spinal cord, and will form spinal ganglia, form neuroblasts, which eventually form spinal ganglia. We have our tube, an inner part of the tube, inner membrane, limiting membrane, and an outer limiting membrane, and a neuroepithelia. So what we have is a neuroepithelia. So what happens? I have said the ventral part is induced, ventral half of the neural tube is induced by secretions from the notochord, sonic hedgehog. The dorsal part is induced by secretions from the ectoderm, the bone morphogenetic protein. So there is a rapid growth of the dorsal part and rapid growth of the ventral part. But before that, we have the neuroepithelia, and this neuroepithelia is pseudostratified. And by definition, pseudostratified is where we have cells resting on a basement membrane, but all of them do not, the nuclei at various levels. So this is what we note here. The cells close to the lumen, the area close to the lumen is what we call the matrix zone. The matrix zone is the area where the cells multiply very fast and they migrate outwards. So as they multiply, they undergo what you call critical mitosis. Critical mitosis. In the critical mitosis, the more differentiated cells remain at the center, but their processes still extend to the outside. The less differentiated cells, again, daughter cells, multiply and the process is repeated, so that we now have a big population of cells occupying the middle part, and those ones close to the lumen as the matrix, still undergoing mitosis. And the process continued to the outside. So we developed three regions. One, matrix zone close to the lumen. We have the mantle zone. The mantle zone are those cells which are more differentiated. And then the outermost layer is what we refer to as the marginal zone. So we have the matrix, the mantle, and the marginal zone. The mantle zone comprises of cells which are not undergoing mitosis anymore, but are now differentiating to form either neurons or gliocytes. Neurons or gliocytes. The first cells to differentiate are the neurons. And then we have gliocytes. But during the migration of these neurons, we have what we refer to as radial glial cells. Radial glial cells. The radial glial cells extends from the matrix zone all the way to the marginal zone. And they are very important in acting as a scaffolding for the migrating neurons. So the migrating neurons attach to them as they migrate out from the central canal or the ventricular surface. So those are the three regions that we have. In the case of the spinal cord, the cells that remain after all the cells have migrated, those we refer to as the ependyma. They line the ventricle, the central canal, in the case of the spinal cord. And the ependyma have no basement membrane. The cells that form the mantle zone, the mantle zone cells now form a gray matter. And then the marginal zone cells form a white matter comprising of axons. I've said the dorsal part is stimulated by bone morphogenetic protein from the ectoderm, the ventral part by a sonic hedgehog. So there is a rapid growth in the dorsal part and a rapid growth in the ventral part, creating a depression. So these cells in the dorsal part and these cells in the ventral part separated by a depression. This depression we refer to as the sulcus limitans. Sulcus limitans. This is what I was illustrating, but this for the brain, sulcus limitans. So the dorsal part, these aggregates of cells in the dorsal part will now, the neurocrest cells here, the neurocrest cells that have not moved very far, that form the spinal ganglia grow a central process towards this part that we refer to as the ala plate. So this is the ala plate, this is the basal plate. Here, the neurocrest cells grow a central process towards the ala plate and a peripheral process extending to the dermatome. So this becomes the sensory part. So the ala plate is the sensory part, and that becomes the sensory root. From our basal plate here, we have very close the somite containing the myotome. So the myotome secretes substances which attract the cells within the basal plate to grow a process towards the myotome. So that process towards the myotome then connect to the myotome. So this becomes a motor plate. So basal plate is motor, the ala plate is sensory. And like I said, this process of the myotome, going to the myotome, when the myotome migrates, it migrates with each segment. And like we had said, each somite contains a myotome. This is what happens in the case of the spinal cord. What happens in the case of the cerebral cortex and cerebellum? We have what is called inside-out layering. Inside-out layering. Just the same neural tube we have with a neural epithelia. We have very close to the ventricle here, the matrix zone, where the cells are rapidly multiplying. We have development of the radioglial cells that extend all the way from the ventricular surface to the outer part, outer limiting membrane. These cells, the first group of cells that migrate, will form a cluster in the intermediate zone. So we have a matrix zone and an intermediate zone containing differentiated cells, both neuroblasts and glial cells. Differentiating cells, just like what we had in the case of the spinal cord, with the processes extending. So we had initially a matrix zone, a marginal zone where the processes were extending there, but now we create from that an intermediate zone. The second wave of migration of cells bypasses the intermediate zone and come to lie very close to the outside, forming a cortical plate. These cells in the cortical plate are still able to undergo mitosis. They can undergo mitosis and therefore develop to form the other layers of both the cerebral cortex and the cerebellum. And the first group of cells that form the intermediate zone will form the nuclei within the cerebellum or the cerebral cortex. The basal ganglia in the cerebellum, a cerebral cortex, and then the dentate, fastigial, emboliform nuclei of the cerebellum. Those ones are formed in the intermediate by the first migration of cells. Of importance here to note is the radioglial cells acting as a scaffolding on which the migrating neuroblasts attach to the outer part when there is the inside-out layering. So this is how we have our cerebral cortex having now the gray matter. Once all the matrix cells have moved out, what remains is an ependyma. Ependyma, and then the white matter, and then the gray matter on the outside. So in the cerebral cortex and cerebellum, gray matter is on the outside, white matter is on the inside, and this is coming out by the process of inside-out layering. Inside-out layering. This is our spinal cord. And you can see from here, the gray matter here is on the inside, the white matter on the outside. We have here our central canal, and the central canal is lined by ependyma, which are the remnants of the matrix zone. And then this is from the basal plate. The basal plate, which we have said grow processes outwards towards the myotome. This one here is the ala, derived from the ala plate, that receive information from the spinal ganglia. So this is the sensory root, and this is the motor root, like we had said. Now, from what we've discussed, we have said we have the ala plate and the basal plate separated by sulcus limitans. This is what I was trying to illustrate, that from the spinal ganglia, we have a central process towards the ala plate and a peripheral process to the dermatome. And this continue on as the dermatome, as the body grows, each part of the skin will carry a segment of the spinal cord. Similarly, from our myotome, the myotomes produce substances which attract growth of the axon from the cell bodies in the basal plate. So this becomes the motor root as it continues to supply our muscles. So this continues on until it comes in contact with the myotome and then forms now the neuromuscular junction. Any cell that does not come in contact with the myotome dies off, undergoes apoptosis. Here we have neurocrest cells. Those that migrate very close to the ectoderm will form the melanocytes. Those that do not migrate very far will form the spinal ganglia. And some of them form the sympathetic trunk. As we look at the autonomic nervous system, we are going to see that. And then the adrenal medulla, we have those that form the enteric nervous system, intramural ganglia of the organs, like we had talked before. All these are from the neural crest cells. Neural crest cells. Now, how then do they connect? We talked about visceral and somatic parts. Here we have the enteric nervous system. We have the ganglia. Then within our spinal cord, or within our brain structure, we have the ala plate and the basal plate. Between the ala plate and the basal plate, this is the area where the visceral supply, the visceral part supplying the viscera. So the upper part, which is ala, is sensory, receiving information from the viscera. The lower part, immediately below the sulcus limitans, sends the information to the viscera. So this part is dealing with autonomic nervous system. The most upper part of the ala plate is purely sensory, purely sensory. And the most ventral part of the basal plate is purely motor. During this growth, we have here a floor plate and we have a roof plate. Floor plate and a roof plate. So again, we are going to see what happens to that floor plate and the roof plate. Well, that's just to illustrate what you were talking about before, rhomboid fossa and so on. What is the fate of the alar plate and the basal plate in our neural tube? How does it develop? Here, when you start with the spinal cord, we've already talked about the dorsal part being sensory, the ventral part being motor. But when we talk about the medulla oblongata, remember the brainstem contains cranial nerves. Within the rhombencephalon, the roof plate becomes very thin, very, very thin, so that our tube now is like an open book, where we have our sulcus limitans here, sulcus limitans. So the whole of this is the alar plate, the whole of this is the basal plate, alar plate and the basal plate. This in the entire brainstem, that is the myelencephalon, metencephalon, and mesencephalon, this contain cranial nerve nuclei. Each of those cranial nerve nuclei could be originating from the alar plate or from the basal plate, and specific parts of the alar plate and the basal plate. Those close to the sulcus limitans, like I've said, are to the autonomic nervous system. But what we have also here is the structures that develop from the pharyngeal arches. Pharyngeal arches. So we have our hypoglossal nerve, which is purely motor. You can see the location right at the extreme part of the basal plate. We have our accessory nerve. Again, motor, but close to the visceral part. We have our vagus nerve. Notice that the vagus nerve has three origins. Three origins. Originating from this part here, which is sensory, originating from what is appearing here as blue, which is motor. So it's motor. That means it sends efferents, the heart, efferents to the digestive tract, and so on, efferents so that our stomach can contract, our heart can contract, and so on. So that's what we have here. We have our glossopharyngeal, and if you notice, glossopharyngeal also has three origins. So they originate from three different types of nuclei, just like our vagus nerve, the salivatory nuclei, and so on. This is part of the autonomic. Glossopharyngeal nerve supplies our parotid gland, and therefore we have that as being autonomic in this position here. Glossopharyngeal nerve is also sensory. So this is purely sensory to our pharyngeal region, anything in the pharynx. As you move to metencephalon, you can see a similar arrangement of our pharyngeal nerves. We will revisit this. As you move to the mesencephalon, you can notice that all our we had what you call corpora quadrigemina, inferior and superior colliculus. The superior and inferior colliculus, when you look at this, originate from this part here, which is sensory. Sensory. So that is for sensory information. It's a sensory relay center. Our substantia nigra originate from this part here. And then if you move to the cerebral cortex, the thalamus and the thalamus, you can see that thalamus sensory is a sensory relay center for all sensory information, be they from the autonomic part or somatic part. All of it passes through the thalamus. And then the cerebral cortex, the basal ganglia. Basal ganglia, you can see, originate from this part here. The cells have migrated ventrally to form the basal ganglia. So we'll be revisiting this when you deal with the cranial nerves. But I wanted you to bear in mind that we have this arrangement in terms of the fate of the ulnar plate and the basal plate. This we have, I think, looked at in terms of inside-out layering, where we have white matter on the outside, in the case of the spinal cord, but in the case of the brain, cerebral cortex and brain, we have white matter on the inside. Now, this one is again just to illustrate what we have as at the most apical part, we have a roof plate becoming highly attenuated. And then we have these cranial nerve origins, what supplies what. Some of these, in addition, we have what we call brachiosensory. These are structures, and brachiomotor. These are structures derived from the brachial arches, the brachial arches. So this is, again, we'll revisit this when you are looking at the sensory systems. That is about the development. Just to summarize, we had induction, formation of the neural tube, regionalization, and also the specification. Craniocaudal, we talked about about homeobox genes. Dorsoventral, on the dorsal part, we talked about bone morphogenetic protein. On the ventral part, we talked about sonic hedgehog. Specification of the cells after migration, proliferation and migration, connection and selection. I brought this out because we need to look at what can go wrong. What can go wrong during the formation of the neural tube? Suppose there is no induction. If there's no induction, there'll be no neural tube. Suppose we have a problem with the formation of the neural tube. Failure of closure of the anterior neuropore or posterior neuropore. What happens? Failure of the formation of the secondary part of the neural tube, the tail region. What happens? We have differentiation of cells. In the differentiation of cells, the first cells that develop are radial glial cells, neurons. These neurons develop first, then you have the radioglial cells which develop. The radioglial cells then make sure that the neurons are the first ones to develop. After that, the formation of the neurons, differentiation of the neuroblasts, then is switched off and glial cells are formed. So neurons are the first to develop, then glial cells are formed. Those radioglial cells now differentiate to form either gliocytes or neurons. Gliocytes or neurons. Suppose we have a problem with migration and proliferation, inadequate proliferation or migration which does not reach its terminal part. What happens? So let's look at anomalies of the nervous system. What can go wrong? Primary anomaly, if you have failure of the anterior neuropore to close, it normally closes by day 25. We have what we call anencephaly. Anencephaly. The brain fails to develop. That part of the brain fails to develop. And then if you have the posterior part of the neuropore not fusing by day 27, then you have spinal bifida. Spinal bifida. This is normally in the lumbar region. Spinal bifida. During the spinal bifida, the vertebral arch also fails to develop. Vertebral arch fails to develop. When you have secondary neurulation problems, the spinal bifida or character can develop where the vertebral arch is missing, and this is normally seen as a tuft of hair just around the sacral region. tuft of hair around the sacral region, or sometimes during the differential growth of the vertebral column, you can have the spinal cord being attached to the coccyx. It doesn't lengthen. So it attaches to the coccyx, so that we refer to as a tethered cord. In this tethered cord, therefore, part of the caudal part of the spinal cord will be stretched, and that stretch will cause damage, mainly affecting bowel movements. Or you might have caudal agenesis, where the caudal part of the spinal cord does not develop. Other anomalies, I think spinal bifida, we've talked about spinal bifida cystica. And in spinal bifida cystica, we have, you can see this normally at L5S1, and this S1 is an area where we normally have development by secondary neurulation. So spinal bifida is a problem that arises either because of failure of fusion of the posterior neural cord. Spinal bifida cystica, part of it will be where we have the meninges herniating into the part of the arch which has not developed, or we have the meninges and also part of the nerve tissue herniating into that part. So that's what they're calling meningocele or meningomyelocele. Then sometimes we can have a complete failure of fusion, the spinal cord in a very big area. So that we call brachyschesis, failure of formation of our neural tube, so that it's completely open at the back. We refer to that as brachyschesis. Syringomyelia, where we have a dilatation of our lumen, And then we have problems of proliferation and differentiation, remember? If you have very few cells multiplying, then you'll have a very small brain that we refer to as microcephaly. But if you have too much proliferation of the cells, you have megalencephaly. But megalencephaly might also have problems with the connection. And therefore, it doesn't mean that you'll have a big brain and you are better off. No. You'll even have some CNS deficits. Then we have also migration, failure of migration. If the cells do not migrate from the cortical plate, adequate cells, and then you might not have, therefore, the formation of the gyri and the sulci, so that you will have a flat brain that we call a gyri. Flat brain, no gyri. Or we might have very small gyri, many small gyri. That's why we call it micro. And that is because of failure of the cells to move to the most superficial part and therefore develop in those regions. And sometimes we have cortical organization. Remember, we talked about the cortical plate during the second wave of migration. Those cells in the cortical plate will make selection. They will attach to each other by forming synapses. And therefore, if you have failure of that proper attachments and so on, then that can also be a problem. Polyprosencephaly, where you have failure of the cerebral telencephalon, where you have failure of the telencephalon to develop properly, so that you have a very big, the two cerebral hemispheres come close together, and sometimes you can just have a single cerebral hemisphere, and that is where you can end up cyclopia. Cyclopia means eventually you develop one eye only, not instead of two. Because remember, they are supposed to come from the diencephalon. And then agenesis of the corpus callosum. Corpus callosum connect the two hemispheres, and therefore you can have failure of development of the corpus callosum. The two hemispheres are not connected by commissural fibers. Fine, this will stop there, and I'll invite any questions. If anybody has any questions, please do ask. But before we do that, I'd like us to revisit one slide. And this is, we talked about the wall, what happens to the wall, where we have the neuroepithelia developing and forming either glial cells or forming neurons. However, what happens to the lumen? What happens to the lumen? And in the lumen, remember we have prosencephalon, we have the mesencephalon and rhombencephalon. Prosencephalon, mesencephalon, and rhombencephalon. In the secondary brain vesicles, we have telencephalon having two cerebral hemispheres, and the lumen therefore becomes there the ventricle, first and second ventricle. So this we refer to as the lateral ventricles. And the lateral ventricles are connected together by the foramen of Monro, the ventricular foramen of Monro. Then we have the diencephalon, and the diencephalon, the lumen of the diencephalon becomes the third ventricle. We talked about the third ventricle when we are looking at the hypothalamus. Remember where we have paraventricular nuclei. When we are looking at the hypothalamus, and we said that the pineal gland developed at the roof of the third ventricle. In the case of the mesencephalon, there is very rapid proliferation of the neuroblasts, so that we have a lot of nuclei in the thalamus, a lot of nuclei, in the midbrain, and this results into the narrowing of the lumen that forms what you call cerebral aqueduct. Cerebral aqueduct. And then for the rhombencephalon, we have the fourth ventricle, all of them are lined by ependyma. And then for the spinal cord, we have a central canal. So this is where we have the lumen developing into the various ventricles. And we talked about the roof plate, and also the floor plate. At the most attenuated parts of the roof plate, we have the pia mater invading into those areas together with the blood vessels, and that forms the choroid plexus. Choroid plexus, both in the fourth lateral ventricles, the third ventricle, and in the fourth ventricle, where we have the choroid plexus, and this is where we have the formation of cerebrospinal fluid. So that cerebrospinal fluid, you will notice that in the cerebral aqueduct here is the most narrow part, and therefore if you have further narrowing, fluid might not pass from the third ventricle up to the fourth ventricle because of that narrowing. From the fourth ventricle, fluid gets out and surrounds the spinal cord and surrounds the brain. And that we have two foramina, the foramen of Luschka and foramen of Magendie. Foramen of Luschka and Magendie, where the fluid now comes out and surrounds the brain, so that the brain is cushioned by cerebrospinal fluid. But we look at the details when we are looking at the individual parts of the spinal cord and the brain. Fine, I'll invite any questions. Anybody with any questions, please put them up. This is the third ventricle. Yeah. This is the thalamus, and this is the third ventricle. Then this opens, if you cut the cerebral hemispheres, then you'll have the fourth ventricle. Our corpus callosum, which you said sometimes can be absent. Here we have our gyri and sulci. Gyri are these elevations and sulcus are these depressions. And this, when there is a failure of migration of the cells and proliferation, then we do not have these depressions and elevations. This part here, our Sylvian aqueduct or cerebral aqueduct, because this part here is our midbrain. So we have a cerebral aqueduct connecting the third ventricle to the fourth ventricle. This is where the fourth ventricle is. And if you remember, we said the fourth ventricle is usually formed by the rhombencephalon. And the derivatives of the rhombencephalon will include this part here, which is the myelencephalon, the medulla oblongata, and the pons. The medulla oblongata and the pons. So metencephalon gives rise to the, at the roof, gives rise to the cerebellum. And this is what we have here as cerebellum of the little brain. Fine, any questions? We'll stop there, and in the next session, we'll look at the spinal cord and look at part of the brain in our next overview before we have a practical. I don't see any questions in the chat, so I presume everybody has understood what we were talking about. The timetable should be out so the class reps can pick them up.